Okay, everybody, attempt number 2 at this video. Um, I had to I had to move locations a little bit. So next part is going to look at the physiology of the eye, but what I'm going to do is I'm going to continue with the anatomy portion of the eye because we didn't look at all the stuff in the retina. So let's look at the stuff in the retina. Meaning stuff, I mean the anatomical structures in the retina, okay? So here's what we're going to see is, okay, so remember this is our retina. So this little yellow part is the retina. And then back behind that is the choroid layer. Because we have that right here, right? There's the choroid layer, there's the retina. C layer, R layer. And basically what is going to be shown here is, okay, imagine I took a little section of the retina right there. So this is going to show you that, yes, this pink area here, this is the choroid layer. Remember, that's in the middle layer. So then if we look at this white part, technically that white part would be the sclera. So you can see outside, middle, inside. So I'm just going to move my image ready. Oh, I'm going to move it back down to the corner here. Okay, so we're going to see that this layer then is the retina. Okay, the retina is going to be composed of several different cell types. I'll color code them a little bit for you guys here. Yeah, red, orange, yeah, sure. So we're going to see that this layer of different types of cells here kind of goes back to about there. This is the photoreceptor layer. I gotta remember, just write slowly. So the photoreceptors there are two types. There's cones, and cones are going to register color wavelengths. Okay, so we're gonna have colored light for cones. I remember this is either red, green, or blue because those are the 3 different types of cones we have. So if you're colorblind, what that means is your cones that might register red don't work, or they register green don't work, or blue don't work. And then we are going to have rods. Rods are the other ones, and these are going to register light intensity. What that means is in dark situations, the cones are more likely to send a message. Actually, they're more likely to receive a photon of light and then be stimulated to send a message to the brain to say, hey, it's low levels of light, and we need to discern depth a little bit better, maybe. But when you have bright light, it's the cones that are actually going to be registering, and then they're going to register specific wavelengths of light. Then the next kind of bluish purple layer, let's just make it kind of a more purple layer here. These are the accessory cells. The accessory cells are basically neurons that are going to transmit an action potential towards the cranial nerve. So what we're going to see here is that this next layer is going to be the neurons of the cranial nerve. So remember, that's cranial nerve number 2, the optic nerve, okay? So at least we now know all of the anatomy. I'm hoping I didn't forget anything anatomically. I really don't think I did. Um, it just seemed like there wasn't a whole lot of anatomy on the eyeball. Technically, there's not. It's just kind of more, it's kind of complicated. Okay, but once you label it, hopefully it doesn't seem as bad as you originally thought it was. So let's erase a couple of things here because we're going to use those. So in terms of physiology of the eye, we want to think about basically this notion. We're going to have light. And we want to understand the pathway of light, which is one thing. So let's just talk about the pathway of light. And you guys can write down what is that pathway of light. So light's going to hit the cornea, and then it goes through the anterior chamber that is going to have aqueous humor. It's going to hit the pupil, which is the opening in the iris, in order to hit the lens, okay? And then essentially that light is going to go through the lens, and when it does, light bends. Okay. Then that light that's bending is going to go through the posterior cavity with vitreous humor, and then eventually it's going to hit the retina. So let's do our pathway of light here. So we're going to see that light has to go all the way to the photoreceptors. So it actually has to go past the cranial nerve neurons, as it goes past the accessory cells, and then has to go past, in order to target the photoreceptors. It actually has to hit this pigmented layer here in order to cause the photoreceptors to be stimulated. So that means our photoreceptors get stimulated, and when they do, they're going to cause stimulation in the accessory cells, which causes the stimulation in the neuron, okay, in the neuron cells. And the neuron cells are closest to the front of the eye. So you want to think about this is the back of the eye, and this is the front of the eye. That's what we were talking about in lecture. The fact that you have to have light go all the way to the back layer, almost to the middle layer of the eye, and then the signal has to be sent back towards the front of the eye. So essentially, we're moving our signal in that direction, and then our optic nerve is towards the back. So that means our optic neuron, our neurons of our optic nerve have to actually send the message backwards as well. So it's like really weird, because basically then that means that that's going to go in that direction. And once it goes in that direction, down cranial nerve number 2, it hits the thalamus. And then the thalamus sends it to the occipital lobe. Right? And remember, it's contralateral, so to the opposite side of the brain. So that's our pathway for light. Okay. So you guys can keep that, or you guys can get rid of it, but sure, I'll just keep my pathway of light on there. What I want to show you is how can we regulate or change light. So there's going to be a couple of ways that we want to think about how light is going to come into the eye. So we can look at superficially what happens to light. So one of the things that we are going to think about changing is changing the position. Oh, that's right. I did it before. Well, one thing we're going to think about is how much light comes into the eye, basically because we open or close our eye. So basically blinking, remember, is done by levator palpebrae superioris. I'm just going to abbreviate because I don't want to write all that out. I don't want to write all that out. Which is innervated by cranial nerve number 3. Okay, so when the cranial nerve contracts, your eyeballs or your eyelid opens and you get in more light. When it doesn't send a message and relaxes, that means your eyelid closes and you get less light. Okay. The other thing is you can squint or like wink. Squinting is going to be done by orbicularis oculi. So the orbicularis oculi, remember, is innervated by cranial nerve 7. And I know this is small here, but that's because I want to use this space for something else. So that's also going to help us regulate how much light comes into the eye, basically how much light's coming into the cornea. You can also have here, you can think about the orientation of the eye. And that's going to be using our extrinsic eyeball muscles, which are innervated by several cranial nerves. So we're talking about, remember, CN3, CN4, and CN6, just like that, okay? Now, the orientation using these eyeball muscles are also going to help us make sure that light gets sent to the back part of the retina correctly as well. So then what we want to think about is, okay, so light coming into the eye, like there's superficial things. There's also for a way for us to regulate how much light is coming into the eye and getting transmitted through the lens. So the other thing we can think about, let's just put number 4 right here, is how much light is going through the pupil. So we can think about the pupil size. Okay, so we're gonna have like an A and a B here for pupil size. One is going to be parasympathetic innervation, and the other one is sympathetic innervation. And what you want to think about is when you're in parasympathetic, you want to go through rest and digest. So you want your pupil to be smaller because you're trying to go to sleep. In sympathetic, we want our pupil to be much bigger because we're under stress. So we're going to need more light in case we need to like fight something off or run away. Okay. Now, pupil size is based on the iris. So in this picture, it's the blue part that I'm going to make a little bit kind of green right there. That's the colored part of your eye. So it's pigmented with melanin. And so you have different, we had different types of melanin. There's different types of melanin that are actually secreted in different places. So the eye has slightly different melanin than the skin does, which is why it kind of gives off a different color. So like blue has to do with it being a light melanin, but I think if I remember correctly, it also shines off of something else. I can't remember exactly. Like, oh, I think it was a lack of melanin, actually. You don't actually secrete much melanin, if any, if you have like blue eye color. So if you have brown or darker color eyes, you actually secrete dark melanin and in higher quantities. Okay, green, I don't remember what green was. But I'm going to use green here because it's a nice color. So in parasympathetic, what we're going to see is we're going to have a muscle called the constrictor muscle or the sphincter muscle, which is the closest one to the pupil, and that contracts. And then we have a dilator muscle, and that's the external muscle. So in this case, our sphincter muscle contracts with cranial nerve 3. And this causes our pupil to get smaller. Okay. In sympathetic, we're going to see that that sphincter muscle is small. And what happens is the dilator muscle, which is on the outside, actually contracts and pulls, and it gets bigger. What is the nerve here? It's a sympathetic nerve from levels. Like you don't need to worry about that. You just want to know dilator contracts. And the pupil increases. Okay, so that's going to regulate how much light hits the lens. And then to figure out what this focal length is here or how to make sure that our image ends up in our retina on the back, we're going to have to change the lens. So when we change the lens, this is called visual accommodation. And at the top, I'm going to put parasympathetic stuff. And at the bottom, I'm going to put sympathetic stuff just to keep us aligned with like what's happening. So you kind of think about under parasympathetic conditions, your pupil is going to get smaller and then your lens has to change shape in order to see objects close up. Okay. And this is close objects. You want to think about like you're eating. So the food is in front of your face. You want to see what you can eat, right? You want to see what you are eating. So in this case, we want to remember like one important thing, our lens is going to bulge. And when I mean that, what I mean is it gets more rounded. So we can see it here. What's going to happen is our lens has to get round like that. So if our lens has to get round, then that means these suspensory ligaments have to slacken. And if the suspensory ligaments have to slacken, essentially what we're going to see is our ciliary muscle has to get pushed forward, right? Our ciliary muscle has to get pushed forward. So this means our ciliary muscle actually contracts. I know that's really hard to think about, but in this case, relaxation causes contraction. So this is cranial nerve 3 that does it. And that causes our suspensory ligaments sorry, my pen's having a rough time today, just to loosen or slacken. And that causes our lens to bulge. So what I told you guys in class is remember, like, you're gonna look at your food, and when you eat, your belly bulges just like your lens will bulge in order to see your food. Usually that means that this focal length here should go closer to the lens, but don't worry so much about where that point is. It's more of a physics thing that I'm not worried about you guys knowing. Okay. Then what we're going to see is in sympathetic, this equals objects closer away or far objects. So I like to think about this as like if there's a big black brown thing running at me, like maybe that's a bear. So I don't know what that is. What I want to do is make sure I can see it from that far distance and discern, is that something to be concerned about? Or is like, I don't know, maybe it's a dog running at me with a happy face. If that's the case, then I'm not going to run away. If it's a big snarling bear, then definitely I'm going to run away. So in this case, what we want to see happen is our lens is going to flatten. And so basically your lens, you want it to be smaller shaped like that, okay? So I don't have any other words besides flat. Flattens are pretty easy. In this case, our ciliary muscle, the ciliary muscle is going to relax. So it's the opposite. In sympathetic, you think about like high intensity stuff, but in this case, your eye relaxes, right? In parasympathetic, you usually think about like mellow things. In this case, our muscle contracts. The ciliary muscle here is going to relax, which means cranial nerve 3 doesn't send it a message. And in this case, the suspensory ligaments, what they need to do is they need to pull tight. So let me put this in a different color. So let's just say, actually, let me just erase this to show you guys what's going to happen to the lens instead, okay? So I need my lens to be flatter, more disc shape. Maybe that's what we'll say. It's like a disc shape. So if we're going to do that, then we have to pull on the lens, which means our suspensory ligaments have to be tight, which is what taut means if you're reading the lecture. So that means that I have to relax my ciliary muscle, and technically relaxation is going to pull my ciliary muscle back. This is because it's a smooth muscle. So since it's a smooth muscle contracts and relaxes in really weird ways. When it contracts, it usually squishes in and kind of bulges out a little bit. All right. So that's how we're going to dictate what's happening to light on the back of the retina. And then you want to think about like whatever you're looking at. So if we're looking at, you know, a picket fence, or we're looking at a couple of lines, let's just say, where they get smaller in the distance, what's going to happen is as it comes through the lens, it gets inverted and flipped. So that means the blue will end up being in the front instead, and this red or maroonish color will be smaller in the back here with the black lines going like that. Okay. Then what's going to happen at the retina is, and I should say, actually, what's going to happen here, let me put that back. What's going to happen with the photoreceptors, hopefully this video doesn't end up being like extremely long. I just want to go over what's going to happen with photoreceptors in the light versus in the dark. And I'm sorry for my really crazy photoreceptor drawings. I just kind of made them simplistically. So that's my photoreceptor, that's my accessory cell. And then this is going to be my cell that's going to my ganglion, like that. I just want to show you what's going to happen in the dark versus what's going to happen in the light. Okay. So in the dark, what ends up happening is that you are going to have sodium constantly entering into the cell. And as a result, resting membrane potential is going to be negative 40 millivolts. And because it's negative 40 millivolts, it's going to release neurotransmitters here. And what this does is tell this next cell, the accessory cell, not to stimulate, not to create an action potential. So basically, I did this like in office hours like this and said no action potential if we release neurotransmitters. Which means there's no action potential in the next cell also. When you have a wavelength of light coming in, when you have a photon coming in, what that does is it causes those sodium channels that are allowing sodium to enter to close. So you're actually going to be releasing more sodium because of the sodium potassium pump. Because you're forcing those positive things out, this causes hyperpolarization, which will be negative 70 millivolts. This stops neurotransmitters. I'm just going to abbreviate neurotransmitters NT, and that actually causes this cell to stimulate. So that causes this cell to depolarize. And then that causes the next cell to depolarize, and so you send out your action potential. That's what's happening in the light. And remember, as we're sending out that action potential has to go back towards the front of the eye, and then it has to go back towards the back of the eye to come out of this part, which is the optic nerve, which is cranial nerve 2. So I hope that helps you with like all the eye physiology. I'm sorry if this is a little bit longer, but I wanted to put all of that together. The next part, let's look at the ear really quick.